And as far as I can tell, if you let them know that it's not simply that we're in the universe, but in fact, given the chemistry of it all and the nuclear physics of it all, not only are we in the universe is in us. And I don't know any deeper spiritual feeling than what that brings upon me. I just wanted to leave you with those thoughts.
Over the course of history, humans have hypothesized as to what makes us who we are. We question what makes us different from each other and how we differ from other organisms. In an attempt to answer these questions, throughout the last 200 years, modern scientists have used the early works of ancient philosophers and conclusions from their own experiments to discover and analyze the molecule of life. This in-depth analysis led to the discovery of the structure of DNA. The first step in the mile-long road that was the development of DNA theory only came to fruition in the mid-1800s, following the scientific revolution. This first step can be accredited to Gregor Mendel, an Austrian biologist and the father of biology. Mendel earned this name for himself through almost a decade of research on heredity using pea plants. He theorized that living things such as pea plants inherit traits from their predecessors. For example, if a white and purple pea plant reproduce, the next generation of plants will either be purple or white. Mendel was able to reliably predict the genetics of a new generation and solidified his research within his laws of dominance, segregation, and independent assortment. Mendel's research marked an incredibly significant frontier in our knowledge of heredity, but the question still remained: what caused this genetic variation? He published his findings in 1865, and these findings still hold true up to this day. Then, later in 1879, a man named Albert Kossel began to analyze a type of blood, leukocyte, and found that it included both protein and non-protein parts. He then added on to this by identifying each component as adenine, thymine, cytosine, and guanine. Despite the convincing evidence up to this point, by the early 20th century, there was a mass disagreement among biologists as to which macromolecule was responsible for transformation. Many scientists came to the logical conclusion that proteins were responsible for transmitting heredity information, due to the 20 naturally found amino acids in the human body. These amino acids are the building blocks for proteins. They suspected that these 20 amino acids would allow for many different combinations, which would then lead to genetic variation. On the opposing side of the argument, several scientists ran experiments to test their hypothesis that DNA was the transforming principle instead. These scientists include Frederick Griffith, the duo of Martha Chase and Alfred Hershey, and Oswald Avery. The four each came to the conclusion that DNA was the transforming principle due to their experiments, which each demonstrate DNA-altering organic material. This collaborative research marked an immense frontier, which was then recognized by the entire scientific world. Not long after this was discovered, Erwin Chargaff added on to this knowledge. Chargaff knew that adenine, thymine, cytosine, and guanine were all found in the nucleic acids within our body. As you may remember, this was proven by Albert Kossel. Chargaff then ran experiments to find that the amount of adenine and thymine were always equal, and the amount of cytosine and guanine also matched. This fact held true for all plants and animals, and became later known as Chargaff's rule. However, his knowledge would not really be utilized until years later, once the DNA model was created. With the work of these previous scientists, the knowledge needed to discover the structure of DNA was present by the mid-20th century. At this point, the scientific world just needed somebody to make sense of and put this information together. By the 1930s, new technologies were made to see beyond the capabilities of a regular optical microscope. X-ray diffraction is a type of crystallography that uses crystals to change the direction of radiation. This is important since the movement of the radiation can be measured, and an image can be made at a molecular level. In 1937, a woman named Florence Bell attempted to capture an image of DNA through this process. This was the first attempt of many to capture an image of DNA, which would greatly aid scientists in uncovering the DNA structure. However, she was unsuccessful and cannot obtain a stable image. Her work laid the groundwork for scientists following her, and helped to admit the idea that women were less capable than men when it came to science. 15 years later, a young up-and-coming biologist at King's College created promising images at the molecular level. Rosalind Franklin was her name, and she was eventually successful in capturing a hydrated image of DNA. She did the vast majority of her work alongside Maurice Wilkins, one of her co-workers, despite their relationship being shaky. Franklin named this iconic image that you captured, Photo 51. This marked a massive frontier in the scientific world, as this was really the first time a woman was acknowledged in the field of biology. Maurice Wilkins went behind her back and showed the photo of the DNA structure to James Watson and Francis Crick without Franklin's consent. To this day, no one really understands why Maurice Wilkins decided to do this, perhaps out of jealousy, or maybe he just wanted to help Watson and Crick in their research. Either way, it was clear that it was unethical. This quickly became incredibly controversial, and may be the greatest controversy in the scientific world to this day. This knowledge that Franklin possessed could have been the tool that she needed to prove that women were completely capable of indulging in the scientific world, which was against society's standards in the 1950s. If the pair of Wilkins and Franklin cooperated just a little bit more easily, they may have had their names done in the history books as the creators of the double helix model. With Photo 51, Watson and Crick noticed that the width of the helix remained constant throughout its length. They were able to conclude that the DNA structure was a double helix, meaning that the DNA was made up of two parallel helices intertwined around one common axis. This contradicted some obscure theories. Then, they took into account Chargaff's rule, that in DNA the amount of adenine is equal to thymine, the amount of cytosine is equal to guanine. It concluded that in the double helix, adenine paired with thymine, and cytosine paired with guanine. The fact that the two paired explained why the two were always equal in amount, because one cannot be present without the other. The application of these rules allowed them to build a structure of the double helix that we know of today. James Watson was quoted saying, "At lunch, Francis winged into the Eagle to tell everyone within hearing distance that we had found the secret of life." This moment marked both the greatest frontier in the history of biology, as well as an immense frontier among the entire scientific world. They published their findings on April 25, 1953. 5 years later, in April of 1958, Rosalind Franklin tragically passed away due to the intense radiation that she endured during her work with X-ray diffraction. In 1962, James Watson, Francis Crick, and Maurice Wilkins accepted the Nobel Prize for their discovery, but Rosalind Franklin went unrecognized. However, it is crucial to remember that Rosalind Franklin cannot accept the Nobel Prize. Her work was not any less influential. Rosalind's work paved the way for female scientists, and without her work, we may have never discovered the importance and implications that DNA brings to modern medicine and the scientific world. With this thorough understanding of DNA, modern scientists are able to provide many applications to their knowledge. You can accredit a great deal of this biological research to the Cold Spring Harbor Laboratory. In this laboratory, James Watson conducted a great deal of research and held an executive position between 1968 and 1994. We had the chance to speak with the current president and CEO of the laboratory, Dr. Bruce Stillman, who stated that
the proposal of the structure of the double helix, which was later confirmed by many means, has influenced enormously the whole of biology, and in particular my own research, because I've studied how the DNA is copied in cells, in eukaryotic cells with a nucleus. And we've figured out pretty much the entire mechanism of how that happens.
In the medical world, we have developed new strategies to treat and prevent diseases. This disease research can be partially credited to the CRISPR technology created by the female duo of Emmanuelle Charpentier and Jennifer Doudna. Charpentier and Doudna are two of the most important women in science since Rosalind Franklin. CRISPR won the Nobel Prize in the year 2020, and for good reason. The organization has begun to edit the expression of somatic cells within our body, and theoretically could be used to enhance the capabilities of the human body. In addition to this, humans have begun to learn where exactly we come from, and we now have a better grasp of our evolutionary history. We've successfully completed the Human Genome Project, which is possibly the greatest feat of introspection in human history. Finally, perhaps the most fascinating of all, we have cloned animals. Moral dilemmas are inevitably going to present themselves as these technologies improve. Questions are bound to arise, such as, when is it okay to alter gene expression in a human? Should gene editing only be used to cure diseases? Is it okay to alter genes to express desirable traits that do not directly affect an organism's health? Obviously, there is still plenty of room to grow in each of these frontiers, and there is still a lot of knowledge that needs to be gained through better understanding of DNA. However, it is important that these frontiers do not go unnoticed. Not only does the story display the many scientific frontiers that Watson and Crick shattered, but also Rosalind's work, which paved the way for many aspiring women in the field of science. As James Watson once said, "Nothing new that is really interesting comes without collaboration."